Hi guys, so today we are moving on to the most important part of the chapter Biotechnology Principle and Processes. In that today we are going to discuss about rDNA technology. So what is rDNA? We have already discussed. rDNA stands for recombinant DNA. Okay. So for making this recombinant DNA as well as to obtain the product out of it, what all the different procedures we are following, that is what we are going to discuss. Okay. So that is rDNA technology is the one which we are now going to discuss in detail. So when you are seeing the steps in rDNA technology, it has been listed like this. Number 1 is isolation of DNA. Number 2, fragmentation of DNA. Number 3, isolation of the desired DNA, okay, DNA fragment. Number 4, amplification of the gene of interest. Number 5, ligation of the DNA fragment to a vector. Number 6, transfer of rDNA to the host. Number 7, culturing of the host cell on the large scale. Number 8, extraction of the desired product. And the last process is downstream processing. So as such the things looks a bit difficult, but the thing is very simple. Okay. So now we are moving on to the first step. So one by one we will discuss. Today we are going to discuss 4 steps. How many steps we are going to discuss? 4. Isolation of the DNA, fragmentation of DNA, isolation of the DNA, desired DNA fragment, amplification of the gene of interest, and we can also discuss more too. Ligation of the DNA fragment, transfer of rDNA to the host. So till this we are going to discuss. Okay. So one by one we are going to discuss. So what is the first point, ma? Isolation of DNA. In short I will tell this one and then we will go in detail. Okay. Isolation of DNA. Like for example, I want to isolate a gene which is coding for producing insulin producing gene or any enzyme producing gene. Okay. So I am taking out from a human insulin if I want to produce, or insulin I want to produce, or any enzymes I have to produce. I am taking out the cell first. Okay, from where the insulin coding gene is present. Right. The cell is taken. The cell first, what I have to do is that from this cell I have to take out the DNA, only the DNA. I don't want the cell wall, cell membrane and all is not needed, nuclear membrane not needed. First I have to remove this cell membrane, then the nuclear membrane, then anything which is attached to the DNA like histone protein, RNA and all which is attached to the DNA, everything has to be removed, and only what I have to take it out, the DNA I have to take it out. So that process we call it as what? Isolation of DNA, which is containing my gene of interest. So here insulin producing gene as such I am saying. Okay. So for just understanding. Okay. But insulin we make it in a different way, but for understanding I am saying it as insulin producing gene, the gene which is coding for insulin. So I have to take that one out. For that what I am doing, from the cell I am totally taking out the DNA. So what I have taken out now, the DNA of the cell has been taken out. So that process we call it as what? Isolation of DNA. After taking this DNA out from this cell, what I am going to do? I am going to cut this DNA into different fragments, isn't it? For that I have told you already, I will add what? Restriction enzyme. This restriction enzyme will cut the DNA at specific location and cut fragments will be created. So that process we call it as what? Fragmentation. Okay. Then isolation of the desired DNA fragment. So from this DNA, what I am getting? I am getting many fragments, isn't it? So I am getting many fragments of this DNA. All these fragments I am putting where? Into gel electrophoresis. You have learnt already gel electrophoresis, right? So into the electrophoresis process we are putting. So all these fragments are put into the wells, isn't it? So then current is supplied. So what will happen? The part when you are seeing the DNA is negatively charged, the fragments of DNA will move over this gel and they will occupy different location. From that I will be taking out my gene of interest, any one fragment which is coding for insulin. What I will be doing it is that I will be taking it out. That process we call it as what? Isolation of the desired DNA fragment. For that, what is the thing which we need to do? Agarose gel electrophoresis. So after taking out that gene, that fragment which is coding for the gene, what I am what factor what I am going to use it. So what I am going to do is that with that one fragment, if I go on carrying out the process, it is very difficult, because sometimes that fragment, by chance, if I am failing in my experiment, again I have to isolate the DNA, then I have to again add a restriction enzyme, then again I have to take that particular gene again. So it is difficult. So when I am taking out the DNA fragment, for example, so this is the agarose gel, isn't it? So in this, imagine this is the fragment which is coding for insulin, the gene which is coding for insulin. So I am taking this one out. Okay. After taking this one out, what I am going to do is that I am going to make many copies of this one fragment, the gene fragment. I am going to make multiple copies of this fragment. That process we call it as what? Amplification of gene of interest. Amplifying, making many number of copies of that foreign DNA. Why I want to make many number of copies? Because if with one, if I am going to do it, if that one in my any processes is spoiled, is destroyed, in that case again I have to start from the beginning, which is very much time consuming and it is tedious also. In order to avoid that, what we are going to do, I am going to make many copies of that desired gene or gene of interest or foreign DNA. Many copies I am making. So that process we call it as what? Amplification. It is done by a process called as PCR. We will be discussing polymerized chain reaction. Then after this DNA is taken, what will be the next step, ma? We have already learnt, isn't it? Foreign DNA after taking, we are taking from the vector the plasmid, the foreign DNA is introduced into the plasmid, isn't it? Introduced into the vector, ligated, and thereafter it is transferred to the host. If it is bacteria also it can be transferred, if it is a plant cell also it is transferred, isn't it? So that is transfer of this rDNA into the host. So that is the next process. Okay. So till this we will be discussing today, right? Now moving on to the first process. What is the first process? Isolation of the DNA. Okay, or isolation of genetic material, that is DNA. For isolating the genetic material, imagine I want to isolate insulin coding region. Okay, so what I have to do is that, or any enzyme coding region I want to isolate. Okay, so that is my gene of interest. So in order to do that, first I have to digest the cell envelope, that is the cell wall, cell membrane and all has to be digested, it has to be removed. Okay, so for removing, if my gene of interest is present in a bacterial cell, I will be removing the bacterial cell wall with the help of an enzyme called as lysozyme. So when enzyme lysozyme is added in the medium, bacterial cell wall will disappear, will dissolve, will get digested by the enzyme what? Lysozyme. Now if it is a fungal cell, I will be using what? Chitinase is the enzyme which I will be using. So when chitinase enzyme is put onto the fungal cell, the cell wall will disintegrate, will dissolve. If it is a plant cell, animal cell though we don't have cell wall. So if it is a plant cell, what I have to use? I have to use cellulase. What does the enzyme use? Cellulase. So the cell wall of the membrane, when you are seeing, is being dissolved by using these enzymes. Now what is present? You can find the DNA is present inside the nucleus, but this DNA, when you are seeing, the DNA is containing not only one DNA, the DNA contains in it histone proteins, you have learned, isn't it? So in the DNA you can find proteins are there, along with the protein you can also find some RNAs are also linked with it. Now I want to remove this protein as well as I have to remove this RNA, so that I am getting only the DNA. So first I am removing the wall, cell envelopes, with the help of some enzymes. Okay, learn the enzymes what are given in the books. For particular bacteria it is lysozyme, for fungi it is chitinase, for plant it is cellulase, like that I am removing it. After removing it, the membranes and all we can easily dissolve it. Okay, like for example, if I am adding a soap solution, all these membranes and all will be getting dissolved. Now DNA will be left out. Now this DNA, when you are seeing, this DNA is having not only a single DNA will not be present, with that associated protein and RNA will also be present. Now I have to remove this protein and RNA. For that, what we have to do, we have to again treat it with some other enzymes. For proteins, to remove proteins, we have to treat it with what? Proteases enzyme we have to add. So the histone proteins from the DNA will get separated. Now next what I am going to do, I am going to add ribonucleases. The RNA which is associated with DNA will also get separated. Now only the DNA I will be getting. After all dissolving all these things from the cell, what I will get? I will get the DNA. The DNA I will be getting it as such. Okay. So this DNA I have to take it out, isn't it? So for taking this one out, to segregate it out first from the solution, DNA is present in the solution now. I have to segregate it out. All enzymes we have added in the solution, what is present? That DNA is present. Now this DNA has to be taken out. For this, what I am going to do is that I am going to add chilled ethanol. What I will add? I will add chilled ethanol. Like for example, you see, I have taken one cell, I have put it in a test tube. After putting the cells in the test tube, what I have added? I have added many enzymes so that all these walls, envelopes are getting dissolved. Thereafter, I am adding what? Chilled ethanol. Now, when the chilled ethanol is added, now you can find that DNA is forming a thread-like suspension on the top. Fine thread-like structures will be formed on the top of the test tube. Okay, it will like fine threads. It will be appearing on the top. That is what is DNA is. Now I want to take that DNA out. So in order to take that DNA out, there is a process involved. We call it as spooling. What is the process? Spooling. So when you add chilled ethanol, the DNA appears as what? Threads. You can find here. The DNA, you can find fine thread-like structure like this. It will be present. Okay. From that, with the help of adhesive substance to some extent, or wood or something like that, you just put it into the what is that, test tube, and just slowly you pick it out. Along with that, what will also come? The DNA will also be coming out. Okay. So this process we call it as what? Spooling. What is that process called? Spooling. Okay. Now I am getting what? The DNA. I have collected the DNA. Hope it is clear. I will tell you again. So for isolating the DNA which is containing my desired gene, my gene of interest, which is coding for producing insulin or any enzyme, that from that I have to take out that DNA. How to take out that DNA? First I have to digest all the cell envelopes, adding many enzymes. Then I have to digest what? The proteins associated with DNA. Then I have to digest the RNAs. Thereafter I will be adding what? Chilled ethanol. The DNA will remain on the top as fine threads. That threads are slowly taken out by a process called as what? Spooling. The process we call it as what? Spooling. Okay. If you are just taking what is that? Any stick and slowly putting like this and taking it out, you will get the DNA out, right? So that process we call it as what? Spooling. After the spooling is done, we are moving on to the second process. Isolation is over. Now we are moving on to fragmentation or cutting of DNA into many fragments. Okay. So for cutting of DNA into many fragments, what we have to do? So the DNA is taken out. This DNA has to be cut with the help of what? Suitable restriction enzymes. I am adding restriction enzyme. This restriction enzyme is cutting the DNA into many fragments. So under optimum conditions, we are maintaining proper optimum condition and we are cutting this DNA into many fragments. Okay. So for cutting this DNA into fragments, what is the enzyme involved? Restriction enzyme. After this restriction enzyme is added at optimal condition, means what? Optimum temperature, pressure, everything is maintained. I am getting this DNA, many fragments of this DNA, isn't it? So because restriction enzyme is added, it is divided into many fragments. These fragments are now introduced where? Into agarose gel electrophoresis. Now we are moving on to agarose gel electrophoresis. So the DNA fragments I am taking, and this DNA fragment, after I take it, what I am going to do? I am going to put these fragments which are collected a little bit in 1, little bit in 2, little bit in 3, 4, like that I am putting in the wells. Then what I am going to do? I am going to connect it to electricity. All these DNA, since it is negatively charged, they will be moving under the influence of this agarose gel. They will be moving electricity, they will be moving. Smaller fragments will be moving faster and they will reach distance, much distance they will cover. The larger fragments which are having high molecular weight, they will be remaining nearer to that of the well. Like that, fragments of DNA, we can separate it out. Okay, so that process we call it as what? Agarose gel electrophoresis. So next step what we are going to do is agarose gel electrophoresis. From that, whichever segment is my DNA sequence, what I will do, I will be taking it out. So imagine this is the gene of interest which is coding for insulin, means that DNA fragment, elution. We have learned the process, isn't it? Elution. Yeah. So that with the help of elution, what I am going to do, I am take that fragment out. Right? So this process we call it as what? We call it as cutting of fragmentation of DNA and isolation of the desired DNA fragment. So that is coming along with this. Okay. So cutting into many fragments, isolating the DNA fragment. This is done with the help of agarose gel electrophoresis. Done. Now after taking out this particular segment which is coding for the gene foreign DNA, this foreign DNA with the help of agarose gel electrophoresis I have taken, isn't it? Then what I will do, I will take the vector also, the same restriction enzyme I will add, and in that vector also I will create a cut, and this foreign DNA I am going to introduce it over here, isn't it? In order to make what? Our DNA. But before this, I have to go for the next process called as amplification of the gene of interest. What is the next process? Amplification of the gene of interest using PCR, which means in this agarose gel, the fragment which I have taken out, the gene of interest, I want to make many copies of that gene. That foreign DNA, gene of interest, I want to make many copies. How to make this many copies? I can make it by a process called as amplification. Okay, and it is done with the help of a machine process called as PCR. So what is the next step? Amplification of gene of interest using PCR. Okay. Now what is PCR? PCR is polymerase chain reaction. It is a chain reaction, the reaction which is going to continue. Okay. Until you control, it will be going on, the process will be going on, continuing till you stop it. So that is a polymerase chain reaction. For this, we are using an enzyme. What is that enzyme? DNA polymerase. So this DNA polymerase is an enzyme which is acquired from, we are getting this DNA polymerase enzyme from a bacterium called as Thermus aquaticus. And what is the function of this DNA polymerase? They will help in making many DNA copies, isn't it? So for making this DNA copies, this DNA polymerase, here in order to make this copies, some amount of temperature in this process, the temperature is somewhat high. Okay, but this DNA polymerase can withstand what? Very high temperature. That's why we are taking it, this DNA polymerase, we are extracting it from where? A bacteria called as what? Thermus aquaticus. What is the advantage of this? It can activate, means they remain active during high temperature. They do not get disturbed. Otherwise, enzymes, if you are increasing the temperature, they get disturbed. But this DNA polymerase is thermostable, which means they are not affected by rise in temperature. And from where I am getting it? I am getting it from a bacteria, Thermus aquaticus. And what is the function of this reaction, polymerase chain reaction? I am going to make multiple copies of the DNA. Gene of interest, what I have taken, I want to make it multiple copies in vitro. In vitro means what? In the lab. Okay. For this, there are three processes. What are the three processes? First is denaturation, second one is annealing, and third one is extension. I will explain. Listen. So, I have taken the gene of interest. This is the foreign DNA which is having my gene of interest. This is the region, okay, which is coding for insulin or any enzymes. So, this DNA fragment is present. Any doubt here? So, you have understood. So, that is the gene of interest. So, this gene of interest, double stranded DNA, when you are seeing, in this I am going to go for the first process called as denaturation. What is denaturation? Heating. Denaturation is what? Heating. I am going to rise the temperature of the medium where I am going to carry out this process. That fragment DNA is taken and put it in PCR machine. So, there actually what happens is that the temperature is a bit risen. Now, when the temperature is getting raised, what happens is that the DNA is there now. the hydrogen bond between these two. Okay, between these two, when you are seeing what happens, these two strands are there. This is one strand and this one is another strand. Imagine. So this strands and this strands will separate. What will happen to them? They will separate. So this is double strand. Now when the temperature increases, so you can find this is double strand. When the temperature increases, you can find the strand will start to separate. They will move apart. Okay. So this is lying close by close. When the temperature increases, they will move apart. Now when the strands are moving apart, this one you can see they have moved apart, isn't it? Next process starts. What is the next process? So what is denaturation? You have understood. I am applying a bit heat. Temperature is increased, for which the double stranded DNA, when you are saying, they get separated, single strands of exposed. Okay. Now here next is annealing. What is annealing? I am adding some primers. 2 primers I have to add, because I want to get first what? This side 3' to 5' also I want to get a copy. Here also I have to get a copy. So first I will be adding primers. What are primers? These are some sequences of DNA chemically synthesized, which is put into here. Okay. So what is this? This is just a template, means they are just opposite to the original strand. Like for example, if I explain, you can understand. 1 minute. You see, this is a DNA. Okay. This DNA got separated into 2 strands. In this, the DNA fragment A, G, C was present. Okay. And here also you can find in this side A, G, C is present. Okay. So now what I am going to add? I am going to add primer. So what primer I will be choosing? I will be choosing AGC, isn't it? So for A, adenine always pairs with thymine. Guanine always pairs with cytosine. Cytosine always pairs with guanine. Here also the same. Cytosine always pairs with guanine. Guanine always pairs with cytosine. Adenine always pairs with thymine, isn't it? So I will be adding primer. This is a primer and this is a primer. This primer will correctly go and join where? To the complementary, to the opposite thing. So A, G, C, when they are seeing, just they are going and joining. TCG is going and joining. And here also you can find TCG is going and joining. So these primers are added in order to start the DNA polymerization reaction to make copies. So these primer, this is a primer and this is a primer. Okay. So that is added. Now what will happen? This primer will go and join. And in this medium, you are having many nucleotides. Nucleotides means what? Adenine, guanine, thymine, cytosine, like that. Chemically, many are there. Okay, already present. So these primers will start the DNA replication process. The next complementary what and all is there will be going and joining. Here also they will be going and joining. Like that, how many will be formed? 2 strands will be formed. Like this, how many cycles it is going to do? 30 cycles. 30 cycle means from 1 to 2. 2 to 4 DNAs will be formed. From 4 to 8. 8 to 16. 16 to 32. Like that, it will go on dividing till 30 cycles. We are allowing them to multiply. So how many DNA fragments will be formed? From one DNA fragment, the same copy, around billions of copies will be formed. Okay. So this is what we call it as what? Polymerized chain reaction. So first is annealing, adding primers. Next process is what? Extension. What is extension? After the primers goes and attach itself to the segments, thereafter the DNA is getting extended. The DNA is formed over here. So that process we call it as what? Extension. So the whole process we call it as PCR. Okay. Now I think you have understood. If you have not understood, once again I will explain. Listen carefully. So imagine this is a DNA, which is foreign DNA containing foreign DNA which is containing some segment which is coding for particular character, insulin producing. Like first is what? Heating. So when it is heated, what will happen to the DNA? This DNA will separate out, isn't it? They will become away from one another. Okay, bands are broken. So this is having some sequences. Imagine they are having sequences A, G, C, T, A, G, C, like this. and this one, what they will have, if it is A here, then it should have been T over here, isn't it? If it is G here, it should be C, C, G, T, A, T, G. If it is G over here, means here it will be what? It will be C, and if it is C over here, means here it will be G, right? Okay, like this the strands have separated. This process we call it as what? Denaturation, heating, right? Now what next we are going to do is that annealing. What is the next process? Annealing. I am going to add some primers. Like for example, I am adding T and C, okay, as well as I am adding A and G. This T and C will go and join where they are trying to join over here, right? And A and G is joining where it is coming and joining over here. So these are the primers which are added, okay, complementary pairs are added. Now what will happen? They will go and join at particular places where there is complementary thing, there they will go and join. So this process we call it as what? Annealing. What is this process called? Annealing. What is the next process? Next process is extension. So in this you can find I am adding many nucleotides, okay, many nitrogenous bases are present. So many nitrogenous bases are present. So what and all nitrogenous bases will be there? You can find G is there, C is there, A is there, T is there, okay, G is there, again C, G, like this many will be there. So AG is already present over here, primers has been added. So next is what? G here, isn't it? So in this G, from here in this medium, what will go and join? C. A means what will go and join? T. T means what will go and join? A. C means what will go and join? G. G means what will go and join? C. Like that, many primers from here again, many nitrogenous bases from here also will go and join here, isn't it? T and G is here, but for T, what will go and join? A will go and join. C, G will go and join. G, C will go and join. A, T will go and join. Like this, you can find this primer is starting and the nitrogenous bases are getting extended, and like this, you are getting how many copies? A, G, C, T, A, G, C, T, A, G, C, T, C, G, A, T, G. Like that, you are getting one copy. Here also the same, isn't it? So A, G, C, T, A, G, C, T, C, G, A, T, G, C. So like this, one copy as well as one copy you are getting from what? One copy you are getting two copies, right? So from this two copies, again the process, 4 copies. From 4 copies to 8 copies. Like this, 30 cycles are repeated and you get billions of DNA fragments. So this process we call it as what? Polymerase chain reaction. Hope this is clear and you don't have any doubts regarding the same, right? So we will just discuss what we have already discussed, then we will learn, okay? So first is isolation of DNA genetic material. So I am digesting the cell wall, and then cell walls and all are cleared, DNA is taken out. In this DNA, histone proteins and RNA is removed, and when chilled ethanol is added, fine thread like structures are formed on the top. From that, by process called as pooling, the DNA is taken out, okay? Then adding restriction enzyme. The next point what we will do, we will add some restriction enzyme. DNA fragments will be made. These DNA fragments are collected and put in what? Agarose gel electrophoresis. All these fragments will move on agarose gel electrophoresis. Only the gene which is I want, that one I will be taking out by a process called as elution, isn't it? So after taking this foreign DNA, I will go for amplification. What is the process? Amplification. For amplification, what is the thing which we are using? We are using PCR, isn't it? So what is PCR? You know, polymerase chain reaction. So the gene of interest is taken, heated, denaturation, 2 strands are separating, primers are added, annealing, primers are going and joining to the complementary base pairs. So after they go and join there, next process starts extension. This will form its own copy, this will form its own copy. So from 1 I am getting how many? 2 I am getting. So like that, many cycles are repeated so that I get many number of gene of interest. So this process we call it as what? Amplification. So for that, an enzyme is responsible for this. The enzyme name is what? DNA polymerase. So what is the name of the enzyme? DNA polymerase, which is produced from where? Thermus aquaticus, okay? So chemically we are synthesizing oligonucleotides are nothing but the nitrogenous bases with sugar and phosphate groups. Okay, so like that I am making many number of copies. So this process is repeated and amplified to billion times. You see, 1 billion copies are made of the gene of interest. So this process we call it as what? We call this one as amplification. After this, what is the next step we have discussed? So we have discussed isolation of DNA. We have discussed what? We have discussed fragmentation of DNA, isolation of the DNA interest, and amplification of the gene of interest. Next is what? Ligation of the DNA fragment into a vector, isn't it? So I have collected a plasmid vector, the same restriction enzyme I have added, and I have made a cut. So there I am introducing my gene of interest into it. One gene I am taking and putting it over here, ligating. Okay. Now what will be in the next process? Transfer of rDNA to the host. For transferring this rDNA to the host, like for example, if I want to, I have made this rDNA, isn't it? So this rDNA to transfer into the host. If I want to make insulin, so I can put it in what? A bacteria, isn't it? So this bacteria can make many copies. All these bacteria will be producing what? They will be producing insulin, because what is present in them? The insulin coding gene is present in them, isn't it? So like that, any enzyme coding gene is present means in the bacteria I will put. But if it is pest resistant variety I have to develop and all, I will put in the host will be the plant, isn't it? The seeds, embryonic stage I will be introducing, or meristematic regions I will be introducing. So for transferring the rDNA to the host, we have learnt competent host. What are the different processes? With the help of chemicals, isn't it? Divalent calcium ions, or with the help of microinjection, or with the help of biolystic method like gun. Okay, so that can be done, or with agrobacterium tumefacium vector, that is disarmed pathogens. So these are all competent hosts which we have already discussed, isn't it? So with the help of all these methods, the rDNA is introduced into the host. It can be chemical, it can be physical, or it can be biological methods. Okay, so with all these processes, the rDNA now is introduced into the host. Next, culturing of the host cell. We will be discussing it in the next class. Hope it's clear. Thank you.